like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Hello and welcome to the National University of Singapore Yonglulin School of Medicine's Healthy Longevity webinar. Thanks for joining us tonight. We have a great speaker, Pura Munoz-Canoves, who will be joining us in a few minutes. I want to remind you during her talk to use the Q&A function to send questions, and we'll try to get to as many of those as possible. Today, we have a PhD student in the Healthy Longevity Translational Research Program, Liu Chong, who will be telling us about calcium transporters and aging-related cardiovascular diseases.
Thanks for the kind introduction. As mentioned, my name is Liu Chong, a first-year graduate student working in Prof Pura lab. Today, I'm going to share my project with you. Before I start, let's look at a set of pictures. What can you identify? Wrinkles, gray hair, and age spots. These are all obvious changes in our body when we become old. It's obvious and we can easily identify. However, there are some more significant changes inside our body. For example, cell senescence. Cell senescence is a process by which cells stop dividing and become irreversibly arrested in the cell cycle, which means the cell cannot grow. It cannot replace themselves. These cells can secrete senescence-associated secretory phenotype, which can disturb the calcium homeostasis. Calcium is an essential mineral. It can function in both the muscle contraction and nerve function, not only for a strong bone. Disturbance of calcium homeostasis can induce many other pathologies in our body, for example, cardiovascular diseases. It's one of the most common diseases affected by the age, accounting for nearly 39.6% of all other diseases. Fukuda et al. Have shown that in 80-week-old WKY and SHR rats, there are less CAV1.2 expression when compared with their 8-week-old counterparts. And the immunostaining results have the same conclusion with the Western blot. Liao et al. Have also shown both the mRNA level and protein level in 16-month-old SHR rats and WKY rats is lower when compared with their 3-month-old counterparts. These results indicate that aging can affect vascular L-type calcium channel expression. Previously, our lab have found that galectin-1 overexpression can reduce the high blood pressure in SHR rats, while galectin-1 deficiency can induce higher blood pressure in mice. Besides, galectin-1 deficiency can also increase the CAV1.2 expression. Based on this information, we can say aging can lead to cell senescence, which can disturb the calcium homeostasis by affecting the function and expression of CAV1.2. Our lab have found that galectin-1 can interact with CAV1.2 And by targeting on the interaction between Cav1.2 and galectin-1, we can regulate the blood pressure. We all know that blood pressure is regulated in either the cardiovascular system and the neuronal system. However, we are still not sure whether it is heart or the brain that plays a more important role in the regulation of this blood pressure. Thus, we hope to analyze the expression of galectin-1 in either the heart or the brain, trying to develop it into a possible biomarker for age-related cardiovascular diseases. Most importantly, our study may find a link between the heart and the brain, contributing to the research field of cardiovascular diseases. Thanks for the support from my lab. This is my presentation. Thank you for your listening.
Thank you. We'll maybe have a future show focusing more on cardiovascular disease during aging. Tonight, we have Pura Munoz-Canoves, a principal investigator at Altos Labs, San Diego Institute of Science. Her research aims to understand how stem cells and tissues age and how to combat their age-related functional decline. In recent years, her lab has demonstrated that at an advanced geriatric age, quiescent muscle stem cells undergo proteostatic failure, leading to senescence, reprogramming their normal daily circadian functions and eventually to regenerative impairment. These studies also uncovered ways to boost regeneration of aged muscles. So the topic of our talk tonight will be improving regeneration of aged muscles. Thank you, Pura, for joining the show. I know it's early for you, so thanks for coming on.
Thank you very much, Brian, for the invitation. Also, I want to extend it to Andrea Meyer, Brian Kennedy, Andrea Meyer. So it's a pleasure to be here. I visited Singapore. I visited your center, I believe it's 5 years ago, and I'm happy to be back even online. So probably this will be the last time I will show this slide. This is a view of my previous center in Barcelona. It was a nice location in the harbor of Barcelona. Yeah, but as Brian said, I moved to San Diego officially in November 2022, but in fact, it was just 2 months ago. We haven't done any experiment yet in Altos. We're just starting, and but anyway, from now on, we'll be here. Yeah, so we study the process of aging as it's so multifactorial, focusing on our tissue of interest, which is over 20 years been skeletal muscle. We have projects on sarcopenia in the lab, but one major angle we study in the lab is muscle regeneration, the formation of new fibers after injury. And we basically use mice as a model system. So skeletal muscle is a tissue with most abundant in our body, but it's a tissue with very little turnover. That means that if no fibers are being formed in normal conditions, its stem cells called satellite cells are usually in a G0 arrest, a quiescent state. They don't need to do anything on a regular basis, on a daily basis. However, in response to an injury, these cells will activate, exit quiescence, expand, proliferate, and form a fiber, which in the end will be indistinguishable from the original one. Like most tissues and organs in our body, the regenerative capacity of skeletal muscle declines with aging, and it is maximal at maximal age. And you can see it here. This is muscle from mice of 3-4 months of age or 28 months of age, what we call geriatric. So, and you can see here that staining for embryonic myosin heavy chain, and that just stains new fibers. So embryonic type myosins that are an indicator of new formation of new fibers. You can see that in young mice after an injury, there are new fibers stained for in embryonic myosin heavy chain. If you measure the size of these fibers 1 week after injury, the size is larger in the young and in the old, but they persist. So in the young, we have more fibers bigger and they persist for a long time. They never mature. So that means that degeneration is persistent and regeneration never is complete, is not completed. Over the years, we focused on this process, and just to make a long story short of past work on stem cells, we found that these cells, the part of the regenerative impairment of very old muscles was because its stem cells had impaired autophagy, which led to the accumulation of proteotoxic products or organelles of protein that were not correctly eliminated in the cell. We more recently showed there were mitochondrial dynamics imbalance, fission and fusion of mitochondria. And all these led to excessive damage in the cell and pre-senescent state of these stem cells. And we found rejuvenating strategies by boosting autophagy, either pharmacologically using rapamycin, that of course does other things, or other intrinsic treatments to rejuvenate the cell. Other groups work on other more physiological treatments, not only affecting satellite cells. So our questions since a few years have been switching from the stem cells intrinsic view, which was right many years ago, but now we know that regeneration also relies on many other cells in the nearby, what it's called the niche, in the nearby this neighborhood of the stem cell, but also on systemic factors. Today, I will want to focus on the understanding what we think are new players in the muscle stem cell niche, in addition to other cell types, and those are senescent cells. So what are senescent cells? Those are basically cells that are damaged. And there's no one good marker alone to identify these cells. They have several traits, and they're larger inside. They do not proliferate, maybe because they have high expression of cell cycle inhibitors. There's positive for senescence-associated beta-gal staining. They secrete factors that may affect the environment, called the SASP, and they are resistant for apoptosis, for example. There are more traits. What is interesting is, besides the increased interest of senescence in the last years in the study of senescence related to aging, related to chemotherapy resistance, most of the knowledge, even today, on cellular senescence comes from in vitro studies. Oncogene-induced senescence, replicative senescence. At the molecular level, most of the knowledge comes from in vitro studies. So that is probably due to the lack of in vivo studies, because these cells are very rare, very, very rare. Even when they are abundant, they're never more than 5% in any tissue. And because the lack of a unique marker, they're very difficult to identify and isolate in vivo. So our question, I go back, is for what I will tell you today, what is the role of senescent cells in skeletal muscle during the regeneration process in young and in aged mice? So we used first this mouse from Judy Campisi, developed by Marco De Maria when he was a postdoc in her lab at the Buck Institute, the P16 3-modal receptor mice, reporter mice, sorry. So under the P16 promoter, there are 3 cassettes, the renilla luciferase, an RFE protein, and a cassette for thymidine kinase from the herpes simplex virus. And in theory, this will be used to, when cells express P16, to detect them with luciferase. Here you can see that after 3 days post-injury, compared to basal muscle, there is luminescence. So after an injury, there are senescent cells, or at least under the P16 promoter. The RedFP doesn't work in this mouse model, and so we had to, I'll link that, because we couldn't detect senescent cells by FACS, for example. And this will be used if we treat mice with ganciclovir to induce the suicide of these cells. So you can see that if here is a representation, we have more luminescence at 3 days, still high at 7 days, and it declines 3 weeks later. If we look at senescence-associated beta-gal, we can see that in young mice, we basically see, or in geriatric mice, only 1 or 2 per section of muscle senescent cells. But in young mice, 3 days after injury, we see senescent cells still high at 7 days, and that disappears later on. However, if you look at geriatric muscle, there are more senescent cells, is the quantification, and they persist longer. Why they persist longer, we can talk later. But those are the kinetics of senescent cells. I am not able to pass. I'm not able now. Okay, so if we now want to see the effect of these senescent cells on the regeneration process, we decided to use the ganciclovir treatment in these mice to eliminate them. This is an experiment of treating the cells for 4 days after starting 3 days after injury, and looking at day 7. And we can do it also starting since day 0 or day 1 after injury. You can see, as I showed you, that embryonic myosin heavy chain staining as a reporter of regeneration in these very old geriatric mice, the size of fiber is enlarged, even if the depletion of senescent cells was never more than 50%. And what was also surprising to see, or at least I would like to call your attention, is that in young mice, we also accelerated the regeneration process measured by this marker. So fibrosis was also reduced after reduction of the senescent cell number, and more importantly, force. More important force. So you compare the yellow against versus the gray bar, you can see more force later on after injury in the regenerated muscle. So pharmacologically, if we look at senolytics, small drugs or compounds that are able to induce apoptosis in senescent cells, what we see using a combination, in this case, of dasatinib and quercetin, there are many senolytics, each week a new one around there. We can see that if we use now a combination of dasatinib and quercetin, we also increase, as with ganciclovir treatment, the size of the fiber. We increase the force. So we can say that removal or reducing the burden of senescent cells is beneficial for regeneration in old and in young mice. How does it occur? So for that, we needed to understand senescent cells or characterize them in vivo. As I said, the molecular analysis of senescent cells up to very recently was only in vitro. So we used a compound that is commercially available, spider beta-gal, similar to C12 FDG, which might be a little bit more widely used. And we tried to, that detects senescence-associated beta-gal, the reactivity. So by fax, we try to be stringent and get the 5% spider most positive cells versus the spider more negative cells. It seemed that what we were getting, if we sort the cells, these are more positive for beta-gal and larger. And we did, from these two fractions, single-cell RNA sequencing. And what we found is that of all the spider-positive cells, the 3 major populations that were spider-positive, supposedly senescent, were fibroadipogenic progenitors, like fibrogenic progenitors of the fibroblasts in the muscle tissue, myeloid cells, and satellite cells, the muscle stem cells. Now that we knew these 3 cell types, we wanted to get further in the characterization. So Those represent three cell types in the niche: the muscle stem cells, the satellite cells, the FAPs, and in fact, if we look like the reporters or markers, PAX7, TCF4 for fibroblast or markers of the three lineages, we can see that the three of them express, for example, P16 in the niche or gamma or DNA damage markers. That's in mouse injured muscle. And the same if we looked at injured human muscle. But what are the molecular traits, as I said? For that, what we did, knowing that the three major populations were myeloid cells, FAPs, and satellite cells, we wanted now to use markers of these cells by sorter and isolate now these three populations to do bulk RNA sequencing, more deep RNA sequencing than the single cell RNA sequencing. So we sorted these three cell types coupling to the spider protocol markers of the three cell types. And we tried to validate if the method was working well. We could see that for the three cell types, we had in the spider positive fraction versus the spider negative fraction, reduced lamin B1 staining, quantified here. We had less proliferation, larger cell area. If we look at telomeres, we saw more accumulation of damage at the telomeres in the three cell fractions in the spider positive. These cells expressed all more beta-gal, and they were not apoptotic compared to the control. So that means that to the best of our that we could, we enriched approximately using 6 markers by 93%, approximately 90% of enrichment using this method. Now we were in good position to be able to isolate this fraction and do RNA sequencing with deeper, with more depth. So we isolated 3 cell types, 3 conditions, 2 time points after injury, 2 ages, 36 conditions, and did attack-seq and RNA-seq. And these are the results. So what the question is, are senescent cells in all conditions similar? Are they, how are they, or different? First surprise was that if we look at all conditions, 2 ages, 3 cell types, 2 time points, and what is upregulated or downregulated or in senescent cells in all conditions, there's nothing in common. No one at the gene level. One other interesting thing was that if we look at what's specifically upregulated, and this is 3-month-old mice, that's senescent satellite cells, FAPs, and myeloid cells, what's upregulated in the senescent fraction of each cell type, we can see that there are things in the senescent cells that are specific for each cell type. The satellite cells, senescent cells of the satellite cell fraction, express myogenic categories at the bioinformatics level. The fibroblastic cells express matrix products, and the myeloid cells express inflammation immune reactive product more than the non-senescent cells. This indicates that the senescent cells still remember what they were. They don't lose their identity. So although we didn't find not one gene that was common to all conditions, if we are a little bit more open-minded and look at the functions that are in common in all the 12 conditions, we can see commonalities like hallmarks or major functions bioinformatically. Many things are commonly downregulated. Many functions are downregulated in senescent cells. And these 4 categories were also upregulated in all conditions. And 2 main ones were matrix remodeling or fibrosis-related functions and inflammatory functions. So we go back to the question now that we know a little bit more about senescent cells. What about the SAS, the secretory phenotype? So for this, what we did was to see what genes upregulated in senescent cells, the transcript coded for proteins that in theory are secreted outside the cells, that they could be part of the SASP. And as you can see, of the secreted transcriptome, if we can say, of satellite cells, FAPs and myeloid cells, they code for different genes, and basically none of them was the same in all of them. Now we are a little bit less stringent, and we look at 8 out of 12 conditions. So the SASP also is pro-inflammatory and pro-fibrotic as the main traits of the senescent cells. But as I say, if we are a bit less stringent and look at the transcripts coding for secreted proteins, that not in all conditions, but in 8 out of 12, we can have 42 genes. And out of those, we could, if we look at in protein-based arrays of the secreted proteins if we isolated these cells and look at what they produce, using these, we could see that there are inflammatory and some matrix-related proteins according to the bioinformatic analysis. What is this? What I'm showing in this slide? It's so what we found is that senescent cells also emerge in the young muscle after injury, and that removing these cells in the injured muscle is beneficial. So we wanted to see how could this be. So this is a comparison of tissues, so different tissues from different species of aging tissues from different species, the African killifish, human, rat, and mouse from published by different groups, which we compared to our senescent versus non-senescent cells in young mice. And we saw that these cells in young mice, if they were senescents, were enriched in similar pathways as these age tissues. To make a long story short, what we think this said that even in young mice, the senescent cells may be recreating by the commonalities with age tissues an age-like inflammatory microenvironment that may be detrimental for regeneration. And that would, of course, this is also conserved in old muscle, but even more worse. So how could this inflammatory profibrotic SASP impact muscle regeneration? For this, what we did was we did a computational analysis taking, in this case, the satellite cells non-senescent, the one that will form new fibers where the regeneration process relies on. And we try to see what factors are produced by the most common, by the senescent cells, and that could impact on receptors harbored by the non-senescent muscle stem cells or satellite cells. And doing these ligand receptor interactions, we looked what pathways could be active within this cell, which could be activated in response to these SASP products or inhibited. Let's focus on what we saw here was cellular senescence was activated by, and probably in a bioinformatically or computationally in the satellite cells, and cell cycle was inhibited. So we try to check that. So here is an experiment in which we use as recipient mice the P16-3 model reporter mice, and we transplant in the muscle of these mice senescent or non-senescent cells. And we can see that the senescent cells transplanted into the into P16-3 mice induce paracrine senescence. Now we can see that the host has luminescence, but this host doesn't have luminescence coming from P16 endogenous expressing cells when we transplant non-senescent cells. So as predicted, we can have paracrine senescence activated because of senescent cells. If we look in vivo at the what's the effect on satellite cells and in proliferative analysis in vivo, we can see that upon removal of senescent cells by ganciclovir treatment, we see increased number of PAX7 positive cells or PAX7 EDU cells that have incorporated EDU in vivo. And different assays told us that removal of senescent cells increases the number of proliferating muscle stem cells. In co-culture studies ex vivo, taking satellite cells and culturing them with spider negative or spider positive fraction, we can see a reduction, even if small, of proliferation of satellite cells when they were in contact with medium from the senescent cells. So the take-home message would be that this was just published. I just have 2 slides. I'll tell you now more of ongoing studies because we haven't done many more experiments. I apologize. So we have obtained an atlas now of senescent cells in vivo to the best of that we could with 90% enrichment. And what emerges is that the transcriptome is very diverse in all senescent cells, but still we have hallmarks conserved. The 2 main hallmarks are there senescents that are pro-fibrotic and pro-inflammatory in their SASP. And this pro-inflammatory and pro-fibrotic SASP has the capacity to induce paracrine senescence on the healthy cells and impair proliferation of the satellite cells. I think we can put senolysis as a way to improve regeneration on very old muscle. The surprise was also in young muscles. And just to tell you that this is not strictly aging, but in models of Duchenne muscular dystrophy, so very severe lethal diseases of skeletal, in which the muscle and the heart is affected because there is the absence of the dystrophin gene that causes a major weakness in the skeletal muscle in the heart, and the constant cycles of degeneration, regeneration, inflammation lead to fibrosis and failure of the diaphragm muscle that sustains respiration and the heart. We can see in this MDX mouse model for Duchenne muscular dystrophy an increase in number. This is the heart, for example, same with skeletal muscle of senescent cells or of fibrosis. And if we reduce for 2 months, twice per week, for example, we've done other treatments. This is an example of 16 to 18 months. If we reduce the burden of senescent cells, even if not yet we have beneficial effects. With that, I would like to propose that we can combine senolysis for some diseases that course with senescent cell accumulation. And these are the members of my group that did the work: Victoria Moisevan, Andres Cisneros, my group collaborators, and the funding issues. And I would like to say that we just put up an issue of cellular senescence on the FEBS journal. There are many articles in this issue covering senescence implicating different processes. So if you want to read more about this, about this topic, it's mainly related to aging and diseases, you can access that. So thank you very much.
All right, thanks, Pura. That was very exciting. It raises a lot of questions as well. And so I want to spend the vast majority of time talking about science, but this is one of the first times we've had someone from Altos. So I thought I'd ask you to tell us a little bit about Altos and why you went there.
Well, I first I never thought I would leave academia. I was happy in Spain. It's my country. And I had a lab that was working well, but Altos represented a new idea of doing science, so we don't have to write grants and we have complete freedom. I mean, I have not been told what to do. Basically, I will continue my research lines. And well, there is a mission in Altos, which is, or a philosophy in Altos, is to work on rejuvenation, trying to use the principles of reprogramming and increasing resilience in cells and tissues. So that's like a mission, and you can embrace it. But in a way, what they want is that the company, what it wants is to do basic research towards finding cures for stopping aging or having healthy aging. And for me, well, the colleagues that I have, it's a very multidisciplinary group, many good people, and a very good funding to work on aging research. And I just couldn't say no. I thought it had all the conditions, and why not to think of an ecosystem working on aging in which there are public universities, public centers, private universities, private institutions, and now also private initiatives that want to do basic science and put the knowledge together there to advance in the fight for aging.
If it's enough to get you out of Barcelona, it must be a really good opportunity. So
ready to answer more questions to anybody, even privately.
So let me talk a little bit about some of the issues that you brought up. And you mentioned that, that aging could be affecting the stem cells, or in this case, the satellite cells or the niche. And, when you did the cell type analysis, it seems like it doesn't completely resolve that, because a lot of the satellite cells are senescent, as well as the immune cells and the fibroblasts. So in fact, I was surprised by how many satellite cells you found in the senescent pool, at least guessing from the FACS analysis. So, or the cell sorting. And so, the single cell sequencing. So, maybe you could comment a little bit more on that. What's driving the decline in repair? Is it the loss of satellite cells because of senescence, or the influence of the satellite cells by the other cells?
Yeah, so maybe I should say that's a very good point, Brian. I should say that that image that I showed, it's not quantitative for the total of cells, because we took 5% of the spider positive and 5% of the spider negative, and they're enriched. What that means is that we equal that. So all I wanted to say, sorry, I went too fast, from all the senescent population, that is still low compared to all the cells. So we don't find more than 5% of senescent cells in the tissue. But out of that 5%, there are 3 major populations. But that doesn't mean that we have lots of satellite cells. But I just, do I explain myself?
No, I understand. I was trying to figure out, because I was going to ask you, where do these senescent cells come from? What's their origin? And it seems like they have multiple origins, the different types of cells. So I was trying to get a sense of how much of it is actually satellite cells senescing, and how much of it is immune cells or fibroblast-like cells.
So I think that, we couldn't say an opinion because this is science. And I think that the loss of satellite cells because of senescence is not the major cause for the regenerative decline. I would say that it is more that all cell types, I mean, I believe that why all the cells in the niche have, based on these 6 marker characteristics, contribute to the overall senescent pop, all of them, despite that these 3 cell types are the major contributors to the overall population. But I would say it's more, regardless of which cell type, what they produce, rather than the loss of stem cells. Because in quantitative, so even if we lose 5% of the cells, of the satellite cells, I don't think that will be the major cause. It's the detrimental effect that they exert on the surroundings. But I mean, yeah, I guess we have many more experiments to prove and be more accurate to your questions. Yes.
It's interesting because at least it's widely commented that senescence promotes wound healing in the skin. And so this would suggest that the role, I don't know if you believe that, but this would suggest that the role of senescence is different in different tissues in response to injury.
Yeah, that's again a very good question, and I mean it. We were so puzzled to see a beneficial effect of removing senescent cells compared to the previous results of Judic and PC. We tried different extents and depth of injury, very mild injury, medium injury, chronic injury. We never saw by a beneficial effect like it has been reported in the skin. And we knew that because it was already reported that we were doing something. We have not in the muscle system seen any beneficial effect of having the senescent cells there. Having said so, we never remove a blade. That's why I was careful to say reduction of senescent cell number and never ablation or elimination. But reduction 50 to 60% of the total of senescent cells benefits the regeneration process no matter what we do. If this is tissue specific, I don't know. But I think that if we are thinking now of all the literature since that original paper in 2014, I would say that there are now more papers in which senescent cells overall is detrimental than beneficial.
You know, the way that thinking about the data, it suggests that senescence, at least in the muscle, is having a detrimental effect post-injury in elderly. So you don't really see a huge increase in senescent cells in older mice that are not injured, I think this we would agree with. And so if that's the case, is this maybe a model for loss of resilience in the aging population? In other words, you can see a decline in muscle with age, but is that a result of accumulation of stresses to the muscle over time, or I'm trying to figure out whether senescence plays a role in normal aging or in the response to damage in aged tissue.
That's again a point that is very important and you notice very well. So when we look at 2 or 3 markers of to identify senescent cells in resting muscle in mice or even in humans, we don't see senescent cells like in other more proliferative tissues. And I'm not really surprised because as I said, muscle is a tissue with very little turnover. So we only see senescent cells that you can say a high number, never too many, but at least a detectable number after an injury. That doesn't mean, so we see 2 per field in an old mouse or an old human by beta-gal. That in a young, you don't see them, but very few in resting muscle. Maybe there is a little bit of them in normal turnover due to daily wear and tear of the muscle, but maybe they'll be eliminated by the immune system, almost undetectable. What we see is that in all mice, we have cells that don't qualify semantically with many markers for senescence, but they have more damage in telomeres already in the basal state. They have more damaged mitochondria. So cells that are a bit damaged, but they're not fully senescent. Or as soon as we do the damage, that's why we have more cells that become senescent, because they were already in a pre-senescence or damaged state. And then they persist longer after damage, probably because also the immune system is not as efficient as in the young mice to remove them. So all this is to say that in resting conditions, we have more damaged cells in the muscle, but they cannot qualify as in the heart. In the aging heart, or in the skin, there is more senescent cells, but in the skeletal muscle, very few. And why then in the studies of, for example, Darren Baker and Jan van Dursen, when they depleted with the INCATAC mouse the systemically senescent cells, the muscle was also benefited, in studies that would be sarcopenia-like studies. Well, probably because they remove senescent cells from many other tissues, and these systemic effects also benefited the muscle. I don't think the effects of senolytics on the muscle at the systemic level, the beneficial effects reported by other groups, are because we're removing senescent cells from the muscle directly, but maybe because of the beneficial removing senescent cells from adipose tissue or other tissues.
So I'm going to take the liberty to ask one more question, and then we'll turn it over to the audience. But I'm really curious about exercise, because it's also a very controversial topic, but there's a lot of people that think, hypertrophy from exercise is due to some low levels of damage to muscle, which stimulates regeneration. You think senescence you can see after a heavy bout of exercise?
Yeah, you can see a few senescent cells after but very few. If it's a healthy exercise, if you do a lot micro damage, you basically don't see senescent cells if it's a micro damage. But if you exceed, and then we enter into, is that good? Then you see senescent cells. But in the treadmill analysis we've done with mice, we don't see senescent cells with exercise. But then other groups claim it, and then we would have to see how we put what are the exercise protocols. Our preliminary data is we don't see basically senescent cells after exercise, but we've done very mild protocols. Yeah.
It's really exciting stuff. A lot more to do, but I think I didn't even get to the muscular dystrophy stuff, which I've worked on a little bit as well. I think it's fascinating as well. But instead of that, I think we'll bring in Sangeeta, who's part of our group, and she's going to ask you some questions from our audience.
Hello, Professor Pura. So I have a few questions from the floor today. So the first question will be, what is the gold standard biomarker in accurately determining whether a cell is senescent or not?
There is no gold standard. If you gold standard within one marker, it's a combination of several. Like it can be P21 or P16 or both, with DNA damage that the cell is not proliferating, and beta-gal positiveness, shorter telomeres, a combination of them. It's a damaged cell, but if you want to call it senescent, maybe you need to have at least 5 markers or 3 or 4 markers. Otherwise, some of these markers are shared by other cell states. Differentiated muscle cell, for example, also expresses P16, so one marker would not, or P21. So we need several markers.
So moving on to the next question. Do we know of any safe baseline number of senescent cells? in the skeletal muscle system that won't negatively affect your overall health during aging?
I don't know if I got the question right, but if we look at 2 or 3 markers of senescence, at least as a requirement to identify, to say we have senescent cells, we don't see bona fide senescent cells in, or a significant number or a detectable number in resting conditions, like in aging. So I don't think that, I'm not talking now about regeneration, which what I focus, sarcopenia, in which you don't need to, so you will only have in a sarcopenia, in a muscle, regeneration, degeneration, if there is an injury. But let's say a mouse or a person that is old, but just has loss of muscle mass and function without any injury. So in a sarcopenia condition, you don't have in the muscle senescent cells in a significant number. So I don't think that doesn't mean that there are not a few. So there are pre-damaged cells, but not, you cannot call them senescence if we're stringent with markers. So the beneficial effects that have been reported and by, let's say, groups at the Mayo Clinic, I believe they may be due to the reduction of senescent cells throughout the body, because those were systemic treatments.
Has anybody looked at an atrophy model?
We're actually looking at in denervation, and I don't have the answer yet, Brian. I can tell you we're trying to do that in models of suspension or denervation, but I don't have the results. We haven't done the treatments to tell you the wrong. Actually, not. I mean, we don't even have permissions to do the experiments yet.
Yeah, so we know how that goes. Sangeeta?
Okay, so the next question. How close do you think we are in finding realistic therapeutic treatments in preclinical or clinical studies that can be used to extend the function of the skeletal muscle system during aging?
What I told you today, and many groups are working in muscle regeneration, this is a particular and narrow field. It's forming new muscle. If we go to what I think your question is like maintaining the health or the good condition of the muscle, this is not, I don't think that has nothing to do with regeneration, because what we want is not to form new fibers, but maintain the good health of the pre-existing ones. I don't think that there are available treatments out there yet. Actually, you have in your institute people, I think that having a very high TOR pathway is bad for the muscle. It sounds counterintuitive, but it is true. So interventions trying to bring down what is aberrantly upregulated in the old muscle or vice versa, playing with pathways that are deregulated in the muscle of the very old mice or at old age compared to young are the right way to go. But for sarcopenia, for cachexia, there are many advancements at the molecular level, but I would say that not yet anything that, even rapamycin has been shown to, even if it reduces muscle mass, but the muscle seems to be better. Well,
so you can imagine senolytics in the context of after injury to muscle maybe being an interesting therapeutic to consider.
Yeah, for after an injury, we've seen that they, at least genetically, or the combination of these two, the dasatinib and quercetinib used, for sure it benefits the speed of regeneration and the overall regeneration in old age. For sarcopenia, for maintaining muscle mass and function, I think this would not do the trick. It will be more trying to see other pathways in the fiber.
Cancer cachexia might be interesting though, because there's a lot of inflammation and senescence going on, at least in the body in that context. I don't know about in muscle, but it's that might be an interesting one to look at as well.
I agree. I fully agree. I think that's a good avenue. We haven't done anything in that direction, but I think it has many possibilities for sure.
So another question, are there any biomarkers that can be used to identify high-risk groups who are more prone to showing a decline in the muscle repair processes during aging?
So people with obesity or with diabetes, as an example, have deficiencies in satellite cell functions and in repair. If you look at the function of the satellite cells in vitro, they do worse even from a person that is not old with diabetes and obesity that are associated syndromes with aging. Yes, and in some models of high fat diet or models of diabetes in mice, the regeneration process is somehow defective. So yeah, I think that those are associated diseases or syndromes to aging in which we can see that the regeneration process is not good.
So is there a sex or gender difference with regards to the mechanism of initiation, progression, or severity of decline in the muscle repair processes during aging?
We haven't looked at sex differences in the repair process. Yeah, compared to sarcopenia, there are differences, but we've studied the repair process and we have not determined the differences. So to say that I am not sure, I don't know if there are or there are not. But this is something really a good question that should be explored, because muscles, there are many traits in muscle that have a gender or sex difference, yes.
So another question, are there any natural ways or supplements one can take to improve the regenerative capacity of skeletal muscles in the elderly?
For the regenerative capacity, we've shown in the past that if rapamycin so DCA mean changing metabolism. If we do pre-treatment, so for example, with rapamycin, we showed that long ago. If we do a pre-treatment of 2 weeks rapamycin in very old mice, and then injure the muscle of rapamycin-treated mice, they regenerate better. We didn't use rapamycin during the regeneration process because that would blunt the proliferation of the stem cell. But if we pre-treated the mice, among the many effects, we showed that we could reinduce or reactivate autophagy whose activity had declined with aging. In other words, we cleaned the cells and made their proteostatic machinery function better. If the cells were a little bit better in their proteostatic activity, and then we did the injury, the regeneration after the injury proceeded better. Yeah. So I would say treatments that affect the proteostatic capacity, in a way, it was inhibiting mTOR. If you inhibit mTOR, which is high, in the stem cells, also in the old mice, if we bring back down these levels, then we can, and other people have shown metformin and Tom Rando has shown that exercise goals are also good for regeneration, which is decelerated in all mice. So, yeah, several.
Maybe, Sangeeta, one more burning question.
Okay, one more burning question. Okay, the question is, did you measure or check the transmyofibrils transmission of amino acids and if any degradation with aging? Sorry, degradation with aging.
No, we haven't done those experiments. I'm sorry, we haven't done them.
Okay.
Yeah, sorry.
Well, Pura, thanks a lot for doing this. A very fascinating talk. I think it's one of the things that to me is exciting about it is it really teases apart some of the different things that happen with aging and damage to muscle and different means that may be needed to repair in different contexts. So I think that's really digging in a little bit more and figuring out what senescence is really doing and what senolytics have the potential to do. So that was fascinating. We'll have you back sometime and hear an update. Thanks a lot. I want to remind participants to use the chat function and the panelists and all attendees option to leave comments so that everyone can view. We're looking for new members of our center, so if you're excited to do that, there's a QR code at the end of the webinar. Also, we're calling for applications for the Healthy Longevity Talent Incubator, which is an intensive course where participants and staff will stay 10 days and 10 nights on campus to learn about healthy longevity science. You can also join us next week, April 20th. Our speaker will be D.T. Gurkar from the University of Pittsburgh School of Medicine, and she will also have an exciting talk. We don't have the title yet, so we'll all be surprised. And we're going to continue tonight on our bodybuilder theme for some reason and talk about an 85-year-old bodybuilder this time. Thanks a lot.
You're never too old to do exercise. I'm 85 and I'm still doing it. A lot of people say, you're still working out. And I mean, I work out 4 times a week. My name is George Campbell. I was born in the island of Jamaica. I spent a little time in the Jamaica Battalion. I was introduced to weight training. You see those guys, so I used to go in the gym, and just fool around. But when I came to this country now, then I go to the gym and start to work out, like a real proper workout, and then I start to compete. I was age 37 when I started to work out. So I started at an old age, but I went for it and beat out a lot of young guys. In the 80s, I was doing 190. Now I do like 165, 170. In the 4 days that I work out, I work the full body twice. Like Monday, Tuesday, I work as one full body workout, and Thursday, Friday, a full body workout, but I do a split routine. I'm committed. When I go to work out, I don't fool around. My motto is a sound mind and a sound body. So that's what drives me. I'm physically fit at my age. I'm a Christian and I read in the Bible that my youth is renewed. I never think of myself as getting old.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.